Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 9th January 2021. As part of our revision, we were revising in three ways. First, we revised the complete revision notes of each chapter with explanation. Second, we revised each chapter under assertion and reasoning type questions. And now we are revising with the important questions. And in addition to that, we are training ourselves in answering objective type questions, 1 mark questions, which play a crucial role in our board exam. As all of you know, repeatedly I am telling you, section A is very important. Section A in coming board exam paper consists of 22 marks. So in the 22 marks, we are answering objective type, which is for 10 marks, and we have already trained ourselves in assertion and reasoning, which is 4 marks. And what is required in coming days, I will train you for the remaining 4 plus 4, 8 marks, that is case study based questions. We'll have a paragraph, and based on the paragraph there will be 5 questions. You need to answer any 4. So that we will train ourselves. So the target is section A. Now coming this session, today we are going to deal with chapter number 10, microbes in human welfare. So we will go for the first multiple choice questions. Colostrum is rich in IgA, IgZ, IgF, IgD. Of course, you know Ig stands for immunoglobulin. Colostrum A, it has to be. Okay, immunoglobulin A. Question 2. Semi-conservative replication of DNA was first demonstrated in. Semi-conservation, of course, we know. I think there is no need. Go straight to D. Escherichia coli, E. coli. Of course, others, Salmonella, Streptococcus, Drosophila, not there. Question 3. The part of the plant used for somatic hybridization is genome, meristem, I think protoplasm, yes, C, protoplasm and nucleus. The 2 microbes found to be very useful in genetic engineering are Vibrio cholera and tailed bacteriophage, Diplococcus species and Pseudomonas species, of course, E. coli and Agrobacterium tumefaciens, Agrobacterium tumefaciens, okay, and D, of course, crown gall bacterium and Cinorhabditis elegans. Of course, we know the answer is C. Question number 5. Physical removal of large and small particle from the sewage through filtration and sedimentation is called primary treatment. You know, physical removal. Yes, this only. And secondary treatment, tertiary treatment, and quaternary treatment, of course, they are all related to biological treatments. So physical removal is primary treatment. Question number 6. Which of the following group of diseases are treated using antibiotics? And here we are talking of group of diseases are treated using antibiotics. Plague, diphtheria, whooping cough. Okay, I think this must be the answer. Diphtheria, leprosy, common cold. Of course, for common cold there is no antibiotic. We will be using the what you call the anti-cold. Okay, leprosy, common cold, plague. Since I think group is A, common cold, malaria, plague. Of course, these are not there. So answer has to be A. Question number 7. In which food would you find lactic acid bacteria, lactobacteria? Mention some of their useful applications, not same, some of their useful applications. Question 8. Name some Indian traditional foods made of wheat, rice and Bengal gram or their products which involve use of microbes. Next, question number 9. Large holes are found in Swiss seas. What is the reason? Give reason. Question number 10. Microbes can be used to decrease the use of chemical fertilizers and pesticides. Explain how this can be accomplished. Question number 11. What is the difference in production of wine and whiskey? Question number 12. Arrange the following in the decreasing order. Most important first of their importance for the welfare of human society. Give reason for your answer. Biogas, citric acid, penicillin and curds. Question number 13. Common yeast is known as baker's yeast and also as brewer's yeast. Justify. Question number 14, very very important. BOD, biological oxygen demand, sometimes it is also called biochemical oxygen demand. Of the two samples of water A and B were 120 mg per liter and 400 mg per liter respectively. Which sample is more polluted? Okay, this is again knowledge based with application. Explain the different steps involved in sewage treatment before it can be released into natural water bodies. This is a major question. So when you analyze these questions, you understand which are the concepts that are very important. Now let's go to the answers. As I said, question number 1A, it has to be IgA because colostrum, also known as bee stings or first milk, is a form of milk produced by mammary glands in late pregnancy and the few days after giving birth to the child. Human bovine colostrums are thick, sticky and yellowish. So the first milk secreted by the mother will be given to the child, and that first milk that comes out of the mammary glands of the mother will be having a lot of IgA. And the colostrum is known to contain immune cells, that is lymphocytes, and many antibodies such as IgA, IgG and IgM. That's why mothers are always advised to give the colostrum containing milk to the baby so that baby's immune system will be very very strong. Question number 2. D. E. coli semi-conservative replication. Not fully conservative of DNA was first demonstrated in bacterium E. coli as it contains a single chromosome within the cell without nuclear membrane. Question 3. Protoplasm. The process by which protoplasts of two different plant species fuse together to form hybrids is known as somatic hybridization, and the hybrids so produced is known as somatic hybrids. The technique of somatic hybridization involves the following: isolation of protoplasts and fusion of different protoplasts. Question number 4. E. coli and Agrobacterium tumefaciens. Microbes are used in genetic engineering technology to alter the gene for getting desired traits. E. coli and Agrobacterium tumefaciens are most widely used bacteria for genetic engineering. Why? They are easily available. Question number 5A, primary treatment. Just now the other time I told you primary treatment of sewage treatment involves physical removal. That's why we said primary. Large and small particles like paper, plastic, pebbles by using screens, through screens. And 6A, plague, diphtheria, whooping cough. Antibiotics are strong medicines that treat bacterial infections. Antibiotics won't treat viral infections because they cannot kill viruses. Antibiotics can treat bacterial infections by killing the bacteria that causes them. That's why the answer is A. Then moving ahead, antibiotics have improved our capacity to treat deadly diseases like plague, diphtheria, whooping cough, leprosy, etc. Next, question number 7. Lactic acid bacteria are found in curds and yogurt. That's why I always insist on you to have everyday curds. And that curds that we eat every day will have a lot of lactic acid bacteria, and they are very good bacteria. They are very good friends for us. This bacteria increases the nutritional quality of curds by increasing the content of, this is the one, vitamin B12. So have curds free of cost, you have vitamin B12. In our stomach, it plays very beneficial role in checking disease causing microbes. Question number 8, wheat product, bread, cake, okay, and of course you have got chapati, puri, biscuits, okay, all this bun. Then rice, of course, idli, dosa, all this. Bengal gram product, dokla, kandvi. So these are the products of the three. Question number 9. Large holes are due to production of large amount of CO2 by a bacterium, Propionibacterium shermani. So this is how large holes are there in the cheese. Next, question number 10. By the use of biofertilizers and biological pest control method, the use of chemical fertilizers and pesticides can be reduced. The fertility of the soil depends not only on its chemical composition but also on the presence of useful microbes in it, which enrich the nutrient quality of the soil. The main source of biofertilizers are bacteria, fungi, and most important this one, cyanobacteria. So instead of putting urea, gromor, okay, you please advise your daddy to go for biofertilizers. And some of the biofertilizers already I have discussed in our tutorials, video tutorials. Okay? I have shown the pictures of it also, right? One of the cyanobacteria, which is the perfect one, perfect 100% nutrition is the spirulina. Okay. And question number 11, wine is produced without distillation, while whiskey is produced by distillation. So wine is an alcoholic beverage made from fermented grapes or other fruits, whereas whiskey is a type of distilled alcoholic beverage made from fermented grain mash. Ingredients, it's made up of grapes. It can be made from barley, malt, and barley, rye, malted rye, wheat, buckwheat, and corn. So many sources. Production process, grapes are crushed into the machine, fermented, and then stored for a period of time. So fermentation process. Barley or other base product is first malted. So these are the differences between wine and whiskey. Moving ahead, alcohol content. Natural wines may exhibit a broad range of alcohol content from 9% to above 16%, with most wines being the 12.5 to 14.5 range. Fortified wines, usually with brandy, may contain 20% alcohol or more, very dangerous ones. Usually contains between 40 to 50%, my goodness. Alcohol, however, it can be made to contain up to 96%. Whether it is 1% or 96%, it leads to death. So alcohol is very, very dangerous, and these are not to be used at all. These are poison, slow poison. It will kill not only the person but the entire family and the society at large. Unfortunately, governments are encouraging this. This is very, very bad governance. So types: red wine, white wine, champagne. Single malt whiskey, blended malt whiskey, blended whiskies, all that. The varieties are there. And then produced countries is giving you Italy, France, Spain, America, Argentina. Of course, we are also producing Scotland, India, Ireland, all this. Okay. And let us not give that much importance to this question. Question 12. The order of arrangement of products according to their decreasing importance is, first one, topmost is penicillin is the most important product for the welfare of human society. Such questions are required. It is an antibiotic which is used for controlling various bacterial disease. Penicillin is the best one. The second most important is biogas. It is eco-friendly source of energy, cheap and best. The next important is citric acid, which is used as a food preservative. So the least important product is curd, a food item obtained by the action of lactobacillus bacteria on milk. Just now we discussed about it. Now come to 13. Common yeast is employed in the fermentation of dough, used for making bread, cakes, and bakeries. Remember Iyengar bakeries? Fruit juices and malt in breweries for the production of alcoholic beverages. Hence it is known as bakers and brewers yeast. 14. Biochemical oxygen demand. I said BOD, or biological oxygen demand also sometimes, is a measure of how much organic pollution is in water. The BOD test measures the amount of dissolved oxygen in water that is used up due to the breakdown of organic pollutants such as sewage in certain number of days. More the value of BOD, I want you to remember this sentence forever. More the value of BOD, more the water is polluted. Hence, sample B is more polluted because the value is more. Why don't you conduct similarly if you got a in your native place, if you got any lake like we have got in Bijapur Botran tank like that, if you got a lake, you can go for calculating the BOD, and you can also calculate BOD in your own well water. You just see and remember the mantra: more the value of BOD, more the water is polluted. So if the values are more in the water that is present in the well of your home, then you understand that you are having polluted water. So take steps to reduce the water pollution. Last, the treatment involves two steps: primary treatment and secondary treatment. This is sewage. Primary treatment, already we talked, it is a physical process of removing small and large particles through filtration and sedimentation. Firstly, the sewage is passed through the wire mesh of screens of sequentially smaller pore sizes to remove floating debris. Then the grit is sedimented by passing the sewage into a grit chamber. The sewage is then kept in settling tanks where the suspended materials settle down to form the primary sludge. The effluent is then taken for secondary treatment. So basically primary sludge is formed in the first one. In the secondary treatment, it's a biological process. The first one is physical process by the heterotrophic bacteria naturally present in the sewage. The primary effluent is passed into large aeration tanks where it is constantly agitated and air is pumped out. This causes the rapid growth of aerobic microbes into flocks, which consume the organic matter of sewage, leading to reduction of BOD. Look at that, reduction of BOD. Just now we talked, higher the value of BOD, higher the pollution. Now here, reduction. After the significant reduction in BOD of sewage, the effluent is passed into settling tanks where flocks are sedimented, leading to formation of activated sludge. A part of this activated sludge is used as inoculum. Like your mummy prepares curds, what she does for making tomorrow's curds, today she will take milk and add one sample of today's curd also, 1 spoon of that. That small dose which she adds into the milk to prepare curds is inoculum, which is pumped back into the aeration tanks. The major part of the sludge is pumped into anaerobic sludge digesters, where in its digestion occurs by the anaerobic bacteria producing very important methane gas, hydrogen sulfide and carbon dioxide. And this combination of these three is biogas. After secondary treatment, the effluent is released into natural water bodies like streams and rivers. The Ministry of Environment and Forest has initiated the programs like GAP, Ganga Action Plan, YAP, Yamuna Action Plan for the treatment of sewage before it is released into the rivers. So this treatment is the best one because it is always win-win situation. There is nothing waste. The primary sludge is used, flocks are used, activated sludge is used, and then finally the water gets depolluted, and in the bargain we are getting biogas. So let's have pure water, and let us put our thoughts into